Welcome to Project AP's November Roundtable. I am Bina Manyar. Many of you know who I am, but it's always good to reconnect on these monthly sessions. Today we have a wonderful team that is presenting some topics that you may have heard about, which I think as caregivers, we all truly recognize as is critical to the health and development of our AP heroes. As scientists, I do think this is a very important topic that we've been trying to better understand with the chromosome 8p disorder. And for those that are providers on the clinical setting at the roundtable, you might find this important and in alliance with a lot of the work that you're doing in taking care of those that are affected by chromosome 8p, and honestly, the extension to the families, the siblings of our AP heroes, mothers, fathers, grandparents, but overall what's happening in the home in terms of what you take in, what you absorb in your system that really gives you the ability to have nutrients to do all of the wonderful things that is so important to live a really healthy life. So today's topic is understanding GI function in our 8p heroes. We have just wanted to kind of take a step back before we go into the agenda and introduce those that are speaking today. Project AP Foundation has come a long way in 5 years. This summer at our Denver conference, we had many families participate in a GI Maps gut project, and that involved many AP heroes, around 30 approximately, that gave their stool to a partner of ours to provide some lab results on that stool. I wanted to share that this is really part one of us better understanding what that means, right? Like kind of looking at our stool, it's one piece of this, we like to say the puzzle often, but it's one piece of the gut. And there is a lot more for us to understand. So today's roundtable is very much this initial discussion around what those results looked like. And overall, we have a larger program of many projects and studies and partnerships that we're doing that fall under, you might have heard, our health canvas. So our Multi-Omics Health Canvas, and GI Maps is a part of this bigger study of understanding what's happening for each of our AP heroes, but frankly, across the community and in collectively. So I wanted you all to know that there's a lot of projects. They all kind of tie in to what today's discussion is about. And I'll share the slide deck. So I just want to make sure you guys can see this. Thumbs up, seeing the screen? Wonderful. So I'll introduce Lauren Chaby for those that have not met Lauren. Lauren is Project AP Foundation's Executive Director. Her first foray into meeting amazing families was actually in Colorado at our Family and Science Conference. So we wanted to welcome Lauren as well. And for those that have not met her, she'll be speaking a little bit overall about what I just mentioned, the health canvas, and how we're using this GI Map stool test to better understand GI. And then talk a little bit about next steps briefly before our host for this roundtable, Geri Brewster, who has done a series of webinars for us years ago in the past, has continued to be an advisor and a nutritionist and dietitian for the AP community. We'll talk about her initial interpretation of the results. She delved deeply into each of the reports for every AP hero and what that means. And she'll also tell you that this is really like part one of it. And then we'll kind of really move into a healthy discussion. We want to hear from all of you about what your questions are overall. And that'll give us as a foundation, an idea of the direction of where we need to go. And we understand that there's always a dire urgency for us to understand this. I'm, first and foremost, mother to my amazing daughter and AP hero. So I want you to know that I share that sentiment. Lauren?
Yes, thank you so much, Bina, for the introduction, and thank you all so much for joining us today. I'm just thrilled to see everyone here and get an opportunity to share some early insights and have this wonderful venue to be able to talk more about the GI initiatives that we at Project AP are moving forward. So this, of course, will seem very familiar, the different genetic subgroups within AP disorders, but I just wanted to come to this to show that this is what one of the many features that we're using to really understand the interface with GI. So we are in the analyses that we'll touch on today and in subsequent analyses, be able to delve deeper into the genetic contributions to GI and keeping in mind these different groupings. So just wanted to flash this here because we're extremely aware of the need to tie these pieces together to really get those insights that we need. Next slide, please. Brilliant. And of course, we're so thrilled that we're able to do this through our IRB approved study. And I just wanted to, again, thank the families participating in this because it's just so meaningful to be able to get together all of our insights under this umbrella and really leverage them to work with the experts in the space like Geri to really move the needle. And we're just so thrilled about the expansions that we have to our natural history study, our health canvas study, and the GI maps being one of the key pieces here and the initiation of our GI work. So we have a little bit of flavor of what that will look like, but just to shout out how impactful this is and how grateful we are that it produces results like we're able to show today. You can pop to the next. Awesome. Okay, so this one will probably also look familiar, but I just wanted to flash this up here because we're focusing in on the microbiome and again to just really demonstrate that it's the overlay of being able to look across these multiple levels of biology and being able to really understand as much as we can about our AP heroes from all of these different levels of analysis, that this is what helps us to really move towards, what are the meaningful subgroups within AP heroes from a therapeutic perspective? What are biomarkers that we can think of within the GI system and more broadly that will help us understand how we're able to make an impact and track this over time in a way that would really set us up for a clinical trial? And how can we leverage all of these insights to think about what therapeutics and opportunities that there are to make an impact for our AP heroes and then be able to measure this? So all of these levels work together to be able to help us achieve those goals. And we're really excited to talk about the microbiome today as one of the pillars of this. Awesome. So the microbiome is of course so complicated, and I'm so glad that Geri is going to be able to address some of this and a tip of the iceberg, I know, even with respect to Geri's expertise, but I just wanted to highlight that, really this is a problem for us all. So about 50% of US adults have at least one condition that could be preventable that's related to diet. And this accounts for a tremendous amount of healthcare spending. So this is really an impactful place for us as a foundation and for us more broadly to be thinking about as an opportunity to intervene to improve health. And this starts with food, which Geri will talk much more about, but I just wanted to flash up here a little bit about the GI system, just because, we talk about it as sort of one lump term, but it is very complicated. There are a lot of organs that are having to function well to really have your GI function well. There's a lot of different components that come into play. And there's a lot of complexity here. And of course, within the 8P ecosystem, we're very used to complexity, but I just wanted to break this down because one of the things that the GI maps test result can do is it can give you some insights about a few of these different layers, but there are some layers that, that are beyond what it can assess. And so this is something that we as a foundation are being really thoughtful about. And we talk a little bit more about this in the next one, if we can pop there. Awesome. So there are many ways to think about measuring GI function. And so one of them is the microbiome composition. And this is sort of the umbrella that GI maps falls into, but GI maps is a little bit more broad than this, and we'll talk about that. But there's other ways to think about this too. You can think about direct motility tests. So you can think about how well are the muscles functioning? How well are things moving through the GI tract? Really from that perspective of the gut-brain axis versus what's specifically living in the gut. There are a variety of other assessments that you can take that can tell you about your GI function. And so this is something as a foundation, we're also really looking to understand how we can address GI functionality from a variety of different perspectives. So getting into it a little bit, what we're here for today, the GI maps. So the GI maps has quite a large number of different pathogens that it can measure. And so we have some of those listed here, but it's a little bit broader than some of the other microbiome approaches in the sense that you're also getting some of these viruses, fungi, parasites, and you're also, so you're getting a larger number of buckets than you might get with a microbiome approach that's deeper. So it's a little bit broader, but a little bit less deep within those categories is one of the ways to think about the GI maps. And so GI maps, the test is a little bit different from what your provider might run themselves if they were to check on one of these bad guys in your gut, because a lot of the hospital systems these days use something called a culture approach. So they actually have to grow it in a dish based on the sample that you give. But what GI maps does is a little bit different. So they're actually using PCR. So they're looking at the DNA of what is in your gut. And so they're not reliant on having it grow to a certain amount. They're not matching it based off of what it looks like, from the perspective of, trying to match what's called like the cellular phenotype. So it's a little bit more sensitive and a little bit more specific because you're not reliant on growing it and you're not reliant on kind of matching them up visually. And so because it's more sensitive and more specific, it can actually give you a little bit more of a deep picture than some of these culture tests, which are still used quite commonly. We can hop to the next, I think. So this is another way to look at all the different indicators that can come from the GI maps. And so just wanted to flash back to this to kind of overlay those two pieces and how this is different from some of the other tests that are available that might be similar. So as we were chatting about the PCR, it is a better method than the culture methods. And the reason that I say better is because it is more sensitive and more specific. And you also get absolute quantitation, which means that you can compare across tests versus if it's relative, it means that I measure it and it looks like a 5, but I'm not comparing my 5 to anyone else. It's just something that is specific to the instrument that I have. And it might not compare to other labs, but absolute means we can put units on it, we can compare it to other labs that are measuring the same things. This is a really valuable thing from a scientific perspective because it gives us a little bit more ability to understand kind of across tests. But so because of this, because it's a PCR approach, it may not look exactly like what you see in the lab or in another test that your provider would order. And we can, I think we'll pop to just a couple of caveats to keep in mind with the GI maps, which I think are on, oh, yes, okay, one more until we get to the caveats. So the GI maps has been in use for over 5 years. And because it's more sensitive, it may detect something that's no longer present in the gut, or something that that an individual wouldn't be reacting to, but is still present. So it's sort of a separate layer of whether or not someone is reacting to what would be present in their gut. And we'll go one more. Okay, yes. So just as I said, if it's positive, it doesn't necessarily mean that you'll have symptoms. And I'll also say that these pieces, these are taken out of the white paper and the interpreter guide that are provided to your provider alongside of the GI MAPS results. So these information about how to think about the GI MAPS test, both in relation to the tests that they may be more familiar with, as well as just sort of more generally thinking about how to leverage GI MAPS is information that they'll have alongside of your GI MAPS result. And so, of course, one of the other big pieces here is that all kinds of pieces affect your microbiome. The medications, whether or not you've had recent hospitalizations, whether or not you've recently been on an antibiotic, all these pieces, they make, what we call clinical history. And this is a really important piece to bear in mind for the interpretation of the GI map. With what they see in the GI maps to get a better indication as to whether or not something that might be elevated or low would be something, and how to really act on that. So it's important to kind of have all of those pieces together to understand best next steps. And yeah, and then the other piece that I would say is just as we were talking about, that whether or not you're reacting to something, it's important to then have opportunities, because this is one snapshot. Sometimes repeated testing is necessary for different reasons. So next steps, once you have your GI MAPS results in hand, or once they're sent to your provider, which Katie has amazingly done. So just as we said, the provider should have your GI MAPS results. So right now, if you've done it with us, they'll be able to interpret it alongside your clinical history, the medications that their AP Hero is using, and dietary habits as well, and they'll have that interpretation guide to help them. They also have opportunities for support for interpretation. So the test manufacturer has a clinical team, and they're able to access that clinical team for a provider-to-provider consult to help understand how to overlay a little bit more of what your AP Hero has been experiencing and what their clinical context is, to just help with next steps to convert it into a treatment plan. And then, of course, our phenomenal Project AP advisor, Geri Brewster, is also, I'll let her speak to available, but we're just so thrilled that she's a part of the team. And these slides will be shared with you as well, so no worries about jotting anything down. And then again, it's important to maybe think about, this sort of, can be really helpful over time to see whether or not things are chronic, whether or not some things are still present. Because there's a component of the degree to which microbiomes change in younger age groups is higher. So this is something to really bear in mind as we think about these results too, is there can be more change for younger folks. And so it can be more important to have additional time points. Just also a nice note that Medicare can actually is something that Diagnostic Solutions Laboratory, the manufacturer of GI MAPS, has shared with us. So this is quite exciting. And this would be through a direct provider prescription. And there are some additional resources. We'll, of course, share the recording of the current presentation, but there are actually presentations from GI MAPS as well. They have a really nice YouTube channel, and the YouTube channel link is in the meeting notes for when the slides will be shared, that you can access and check it out. They have, like I said, quite a lot of good ones that go through kind of a page by page, as well as some other kind of broader usages of the tests that they've done over the years. And then on to the next. Awesome. And so this will also be shared with the slides, but should your provider want to reach out for a provider-to-provider consult, for interpretation of the GI maps, they would complete this account setup form and then share it with the test manufacturer, the Diagnostic Solutions Laboratory email that's listed here, and the form will be sent along with the recording as well.
Is there another slide, Lauren?
I think maybe.
Let me just check before I. Okay. Oh yeah.
I think it might.
Hold on. There is. Okay.
Thanks, Mina.
Yeah, no problem.
And I'll just say as she brings us up, thank you all for your patience, because I'm in the car on the way to see Mina, and I hit some traffic, and so I unfortunately I'm a passenger in the car, so I'm not driving, but I'm a passenger. Oh yes, okay. So next steps of kind of the GI initiative within the Health Canvas. So we're of course doing a deeper dive into the GI maps, and just as I was saying, we're overlaying it with these different data sets. So this is really exciting. We have a team of data scientists that are working on this and should have some neat things to share. So stay tuned for this. But we really wanted to have this opportunity to talk about, as you guys get the results in your hand, you know, wonderful tips and guidance from Geri, who's been able to dive into every individual. And then, so the other thing that we're doing, and I'm really excited about all of this, but we have a partnership with Clinical Microbiomics, which is a phenomenal group. And so they have one of the best reference libraries in the world for the microbiome. And so we're doing shotgun metagenomics, where we're going to be able to see all of the different components of the microbiome. And so this is going to allow us to dive into that one column that we were talking about that GI maps addresses, but dive into the one column very, very deep. As deeply as modern science can really go. And so this is really exciting. And what we're going to do is we're going to overlay that with as much as we can of the context. So there'll be some surveys that go along with this to really help us understand GI health and the dietary impact in AP. And, you know, just as next steps to really be able to understand what opportunities are in this space. And also, we have a phenomenal science writer who is going to be putting together a Substack article to talk about all of these initiatives and talk a little bit more about what you'll hear today. But we're really excited that this is the tip of the iceberg for what we can do in GI. And I think, you know, what we've seen thus far is that there really is a reason for us to invest in this space. There's a lot of heterogeneity, which isn't surprising, but a lot of variability in what we've seen from the GI maps. But there is that piece of, you know, the very real need here that's represented that I know Geri is gonna cover exceptionally well. But because of that need, you know, we as a foundation are really dedicated to understanding what opportunities are here for us to move the needle to improve health for our AP heroes. I think that might be it. Oh yes, and an invitation. You know, please do join us for our partnership with Clinical Microbiomics. You know, we're just so excited about this. Particularly, I think it's going to overlay with the GI maps data set in a really exceptional way to give us some wonderful insights into what's going on with the microbiome of our AP heroes. And so this will help us, you know, one of the big pieces is it's not just that we'll look for, you know, known knowns, as they say, but we're going to be able to look for the unknowns. So things that we wouldn't necessarily think to look for that might be causing, you know, GI challenges for these kids, we'll be able to see that. So this is going to be wonderful. And I just pulled this from the website of Clinical Microbiomics, but they actually have specialization in overlaying these different data sets. And so, you know, they're going to be able to help us to even leverage more what we've already generated. So this is just, it's a phenomenal opportunity for us, and we're just so excited.
Great, thank you so much, Lauren.
Thank you.
I'm going to switch over to Geri, who has spent a lot of time and energy caring so much about our AP community. And hold on, yeah, there you are, Geri. Okay, great. I'm going to share the slideshow and be the master clicker.
You're
So
doing a great job. And Lauren, that was fantastic under pressure. So thank you guys. What a team, right? And always, well, we're all here together. So, but while you're pulling that up, Bina, I just a couple of notes just to kind of follow on what Lauren was saying and segue. Um, the that cultural component in terms of what some other providers, GIs or pediatricians may have done in the past, often it is looking for ova and parasites, like something very pathogenic, as mentioned. And again, I'm saying this only because when we're in it all the time, the language, even as we're, it's also a teaching model, and we can use words that that are a language we're familiar with. So so when the tests that are often done conventionally or through your practitioner or provider may very well just be looking for the bad bugs and are not necessarily, they're not going to look for or culture out everything else that is there, good, bad, or different. They're only going to even rule out the known bad bugs, the listeria, the giardia, the salmonella, understandably, and that's important. But GI maps and some of the other GI microbiome tests, as Lauren referenced, they are PCR. So yes, they're going to be much more sensitive and much more specific, but also much more comprehensive. So they're telling us everything that's been there and has had a presence. So also, as she said, because it's PCR, sometimes depending on different levels and a little bit of a range sliding scale based on age even, and a developing microbiome, right? The first 1,000 days of life that I touched on in our at the conference in the nutrition segment. So there's presence, and so it's a snapshot, but a snapshot of sorts. It's going to give us a little bit of a look back. So it's a bit of a retrospective as well as what's happening, right? When that particular stool sample was taken. And it has significant influence because everything is operating together. So one marker is going to impact another marker, right? There are downstream effects and so on. And we'll get into that as I go through my slides. So I don't want to really digress too much. And most certainly all of your providers, while we have this tremendous amount of resources and GI maps, besides, yes, their YouTube channel is great, but just right on their main site under even whether you're a provider or not, even as parents, if you want to go in, it's all accessible. All of their various white papers, interpretive guides, short videos, but not everyone can become an expert. I have been running this test for my patients in my practice since the 5 years since the company began. And the learning curve and the literature is forever growing, right? This is still something that has just come to be and recognized. The microbiome, that collection of parasites, fungus, viruses, some that should all be living synergistically together, but certainly some that should not be present, and so on and so forth. So that whole collection from our oral cavity to our anus, everything is impacting. So even chronic dental infections, sinus infections, bacteria, right? We all are familiar with our mouth bacteria and responsible for cavities, those bacteria that can create those biofilms in the mouth. Well, it all goes all the way down. So with that, that's enough, but just to, so your providers can also always call on me as necessary, as well as setting up their relationship with GI Maps to then call on the great consultants and practitioners within GI maps as well. So, all right, so we're on the first slide. We can definitely dive right into next slide. And which I think I probably already kind of covered in my a little bit of my preamble here. I'm sorry, Bina, did we? Yeah, jump back a second. I'm sorry. So some of that already mentioned, and as Lauren mentioned, with regard to adults and the influence of the microbiome, certainly also with children, and knowing that so many children now, significant number, and the CDC is always lagging behind with its publications, but at last written 40% of school-age children with a chronic disease, meaning it's always going to need some kind of health intervention. But many of these have been found to be directly influenced by what's going on in our microbiome. So looking to really optimize the microbiome is significant. We speak certainly of 8P as its own condition, but all of the heroes are also going to be having to manage all the other health concerns that prevail on this planet. So we want to definitely avoid what we can by optimizing the gut. So the next slide, and again, I'll try to go through these quickly so we can run into the details of the test a little bit more. So all of these, the gut microbiota and the studies that have been done and the implications, and again, just like the genomic study, the microbiome study, these are studies that have really just been completed more or less as a body of information in these last 20 years. So, but this is the essence of our health and disease. So again, you'll get copies of all of this, but dysbiosis or an imbalance of our gut microbiomes between basically the beneficial, the known beneficial, and the non-beneficial, right? We're always going to carry a certain amount of non-beneficial in our gut, enough to kind of prime and keep our immune system at the ready to operate in a nice homeostasis to protect us, but that goes awry very often. So that imbalance between good and bad bacteria or dysbiosis can also lead to our vulnerabilities of more pathogens or frequent, GI bugs or other GI disease. And it is definitely associated with many pediatric conditions, as noted in this 1, 2, 3, 4, 5, the dysbiosis, fifth or sixth bullet point there. Those last couple of bullet points especially. And this again is recent out of Journal Gastroenterology, World Journal Gastroenterology in 2022. And then in our own heroes, we also know from what's been published that IgA deficiency, and we'll talk more about IgA, that is that nice immunoglobulin, that first layer, and all the mucus layers from respiratory to gastrointestinal, that first line of defense. But many suffer with low IgA. So we're going to look at that. And then other conditions that have been reported. And many of these conditions reported in the 8P population also are what's seen in terms of microbiome dysbiosis within the general population as well. So again, all right, next slide. We want to really be able to optimize what we can by moving into optimizing what's known as this GALT or gut-associated lymphoid tissue. That is everything that's living in our gut, which should be really that nice, thick barrier protection that allows us to digest and assimilate and absorb the nutrients we need while capturing and eliminating all of the non-beneficial antigens that come in on our food supply from antibiotic residues or pesticides or pathogens, like again, today's headlines on the E. coli in the carrots. And everything that comes along with the adulteration and processing and transportation and mass production of our food supply. So we need even a more robust gastrointestinal tract than ever before. And within the gall live these dendritic cells, which are so significant to maintaining gut homeostasis and therefore preventing inflammation and also preventing the kind of inflammation that leads later on to an auto-inflammatory state or an autoimmune state, because there may be genetic predispositions toward Crohn's or colitis or psoriasis or rheumatoid arthritis. However, the turning on of that genetic expression has everything to do with what's going on in the microbiome. Next slide, please. I also have to keep up on my end. It's like my brain's going beep, 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 because you're.
I see that.
I'm getting into the groove. And also, I'm just kind of looking at it and I'm just speaking to all of you because there is a lot that I do want to share. And I also want to make it understandable for everyone so that there can be a better application and appreciation of some of the dietary changes and just the small things that can be done every day and the influence of those small steps on can have long-term significant beneficial effects, and not only for the heroes, but all of the families at large, because we are talking about essentially a huge public health component, which our 8P heroes are, in fact, we're all living in this kind of dysbiosis and chemical soup environment, unfortunately. Okay. So those dendritic cells, though, and that dysbiosis or that bacterial influence, it can then, as I mentioned, drive our entire gastrointestinal tract into a more pro-inflammatory, creating more inflammatory chemical compounds or these cytokines versus anti-inflammatory. And that's why in that early discussion, as Lauren mentioned, and then I reiterated at the beginning, even though we're looking at this particular stool sample at that time that you took it from your child, it is PCR, so it is giving us a little bit more of that, looking back, as well as what's happening at that moment, and why it's always beneficial then to implement some dietary changes or supports and look at them sequentially so we can move the needle into slow progressive improvement over time to get to that optimization, because there can be setbacks, most certainly a GI bug, a short course of antibiotics, but it's not that we're always swimming upstream. It's just now we need to be aware so that our trend will always be to the better. And we don't have to do the tests all the time, but depending on your child's initial results and how we're going to determine measurability, it may be something, usually we would want at least 12 weeks to kind of look for a true turnover and improvements. But if there is something very acute that needed more immediate medical attention, we may want to look at that in certainly a shorter window of time, just to make sure that some of the downstream inflammation has been taken care of. So, because that strong intestinal barrier can sometimes be broken if there is a lot of inflammation going on. And that, in fact, is what may be referred to really as leaky gut, right? When all of the things that should stay in the gut to be processed and eliminated out, some of them are going to get into our bloodstream because the barrier is broken, the defenses are down, that nice thick mucus layer where all the final stages of digestion and detoxification can occur may be low. And also, whole proteins can then sometimes influence. So it's not just even the bad stuff we don't want getting into our bloodstream, but good stuff you would think, oh, protein, but not in that whole protein form because it's too large. That could ignite a more systemic immune response, right? Which then takes its toll on our kiddos. Next slide, please. And again, I think as I already suggested, a lot of this can be reversed through some of our dietary changes. What's referenced here is that compound called zonulin, which showed up for many of our heroes, and we're getting closer to getting into the details of the test results. And that is a marker essentially of inflammation in the small intestine. It can be triggered by chronic inflammation. It is most certainly also triggered by gluten sensitivity. But of course, gluten is a large molecular protein. So even if you don't have genetic celiac tendencies in your family or in your child's own genetic profile, you can, in fact, wind up still with high levels of zonulin because of chronic gut inflammation. Next slide. So, and again, because of this barrier breaching between homeostasis and inflammation. All right. So this is why, next slide, why from the dietary standpoint, fiber and whole foods matter. As I said to many of the families that I spoke with at the conference, while Miralax may be necessary to help with the elimination, which is very important, it is not going to benefit the microbiome. So we still need to focus on these nutrient-dense foods that contain the fibers and those compounds, these polyphenolic, colorful compounds in our fruits and vegetables that help beneficial bacteria to proliferate in the gut and help maintain that sense of homeostasis and good immune response and good barrier function, and that nice, diverse, healthy, robust microbiome. And as noted here on the top, even these plant-rich foods, everybody hears about GLP-1s now in terms of like Wegovy or Ozempic in terms of regulating motility, gut-brain connection in terms of hunger and satiety, but also GLP-2, which is yet another compound that a well-fed microbiome will produce naturally on its own. A lot of people don't even realize that Ozempic and Wegovy are compounds that are based on ones that we would make in our own gastrointestinal tract. But GLP-2, yet another compound that comes as a result of providing these whole fibrous food, colorful foods into our diet every day, is one that also helps support good barrier function. So there's more to it than just fiber. Okay. So all of that is how food influences gut bacteria, which in turn primes those dendritic cells, which influence the gall to determine tolerance or inflammation, and really then influences your genetics towards health and disease. Next slide. Sorry, I think I did move to my next slide. That's what I just said. Oops, sorry, here we go. I'm also rushing because I know we're a bit on a time. All right, the role of butyrate, a very significant compound to come out of all of these nutrient-dense fiber foods. And we can have another conversation about fiber, maybe in the part 2 of all of this. But as a result, those compounds in those foods feed the good bacteria to produce a multitude of short chain fatty acids. One very specific focused on today is butyrate, because butyrate is something that transcends the gastrointestinal tract. It definitely helps to modulate inflammation, homeostasis in the gut, but it's very neuroprotective for the brain. It's very important for our bone density. and growth, which many children struggle with as well. It is very responsible. Yes, we know from tone issues and specific innervation of nerves into the colon with an 8P diagnosis, there is going to be slower transit. So we must optimize our butyrate production in the gut by optimizing the butyrate producing good bacteria, because butyrate is essential to stimulating those colonic contractions that allow for regular bowel movement. So we want, as I said, sometimes you need the laxative or the Miralax. But at the foundation, we still need to have all the good ingredients going in every day into our children's gastrointestinal tracts in order to optimize the well-being and function of their gut, which is then going to be responsible for their growth and development, their bone density, their neurological and immunological systems. Next slide, please. Okay. So with regard to nutrients, similarly, the gut runs on a lot of nutrients. If we're not getting them from our diet, we may in fact need to provide them supplementally for a short period of time. But also when your gut is functioning well, it's also going to be able to produce vitamins, right? So it's very sophisticated how well it really runs, and it's running all the time, and we take a lot of it for granted. So looking now, next slide, into page 1 on your GI MAPS test, and this is just an example of page 1, which is really a look at the pathogens. The things that should not be there, that you may have heard about. C. difficile. Now, Clostridia difficile can be in our gut at low amounts, but when it overgrows, it will produce toxins that are extremely inflammatory and damaging and result in a colitis. Another example here on this are pathogenic forms of E. coli, and again, they're toxins, the sugar-like toxins that can be associated. The salmonella and the giardia that I mentioned earlier in terms of pathogens, things that really should not be there. So that's that pathogen page. So if we move to the next slide, there were some bacterial and parasitic pathogens from page 1 were identified in a number of the heroes. So there were 29 tests done, and we picked it up on 5 of them. And as I mentioned already about concerns with C. diff, it's a bacteria that's there. Giardia, it's there, but should only be there in certain levels. Giardia is a parasitic infection that should not be there. And again, if the gut is compromised or struggling and it's very inflamed, it's going to be more vulnerable. So that's even when the CDC or your local health department has to declare a foodborne illness, it's only based on 10% of the population that ingested it. So if you go to a wedding of 100 people and 10 get sick, that could be enough to suspect that it was a foodborne illness, even if everyone ate the same food. You don't have to say it must have been a GI bug because we all ate the same food. We would have all been sick, because based on your microbiome and even the amount of your stomach acid, right, to kill the bacteria right away. So our guts have different resilience. So this is why it's good to take a peek to just see how we're doing. So anyhow, with regard to Giardia, it can be somewhat opportunistic. Anything, we can get so many foodborne illnesses through contaminated food and water, and whether we do or we don't has a lot to do with our own gut resilience. Some of these bacteria would be there if you just had a recent, bug, right? A gastroenteritis that could be gone in 24 to 48 hours. It was something. So again, that might be picked up here. All right, next slide. So page two looks at Helicobacter bacteria, Helicobacter pylori or H. pylori, that many people around the world and everywhere live very comfortably with, although it can be problematic even in low levels, and I'll discuss that in a moment. But also what's noted on page 2 of the test are the commensal or keystone bacteria, the good stuff that should be there. Now, there are a number of different phyla that occupy the gut, but the two main houses are Bacteroidetes and Firmicutes. You see that at the bottom of the page. So at the top, we have the Helicobacter pylori, a bacteria that honestly can live with us, but shouldn't be necessarily part of our usual microbiome. But depending on how much of it we have and whether or not we're symptomatic, it may or may not be an issue. The commensal and the keystone bacteria, and then their collective amounts within these phyla, the Bacteroidetes and the Firmicutes. So now if we move to the next slide to just dive in a little deeper there. So quite a number of the 8P heroes tested did show positive to exposure to H. pylori, but many had levels that would not necessarily be symptomatic. So, and symptoms of real H. pylori infection can be significant reflux and dyspepsia and crying after a meal, perhaps, because it can lead to chronic gastritis if there's even anemia, because there could be slow, little, if your stomach lining or gastritis is very irritated, there can be some microscopic bleeds going on that over time would lead to anemia and also low energy. But for some, as I said, it can just hang out. It's very communicable. So just even like if you've got it and you're kissing your baby, or it's very communicable. But at some of these lower levels, it wouldn't even be able to be detected with a breath test. And of course, some of our heroes may not be able to cooperate with a breath test. Even when I've done breath tests, there's still a rhythm and a learning curve that's all very challenging. But nonetheless, at the end of the day, it could still influence the pH of the stomach, right? Because it produces urease, and urease will alter the pH of the stomach. So we get less stomach acid, and with less stomach acid, there's less signaling to the pancreas that food is coming. Please produce all your pancreatic enzymes to further digest and assimilate what's coming down the pike, because otherwise it will be left to maybe some opportunistic bacteria that would like to feed on some of that food that is now not getting digested or assimilated into the body of the person consuming. So next slide. All right, so and that's why we look for certainly enough of the good commensal to help keep those opportunistic that we'll look at in a little while in check. And this is how everything's all interrelated. So to try to look at these groupings can, we're looking at them in isolation, but I'm thinking about big picture as I'm going through this with you. So it will be worth a nice review once you get these slides yourselves, and then have the opportunity to just take a peek at your child's with your provider, or once your provider provides it to you before you even have an opportunity to meet with them, or perhaps meet with me, or for them to meet with one of the providers at a Diagnostic Solutions lab, which provides the GI MAPS test. I don't want anyone to, I don't want anybody to get upset when you see some of the skews, because this is why we are doing this, right? Because we certainly know some of the symptoms that are being presented. So now when we take a look inside, we have a better understanding of why maybe there are either certain skin conditions or gut upsetness or bowel issues. So it just gives us a really good starting place to know what dietary interventions and support we're going to begin with, to pair with whatever medical interventions you are going to get the advice from your physicians and gastroenterologists. So these commensal and keystone bacteria, the good guys, as I said, two major phyla, the Bacteroidetes. They start very early on. Those human milk oligosaccharides, those carbohydrates, they are there to feed these first bacteria. And the bifidobacteria, which during a vaginal delivery, a baby will get inoculated from, just coming down the birth canal, and then those human milk oligosaccharides from breast milk and also from formulas it's being added to now, feed that good bifidobacteria. But if if a C-section was necessary or antibiotics at delivery, some of these good bacteria get wiped out right from the beginning. And unless there was recommendations for, say, reseeding, as it's called, with infant probiotics, they may have never gotten a really good, foothold. And then the firmicutes, those bacteria really influence metabolism via that short chain fatty acid production, as I mentioned, the significant one being butyrate earlier. So Faecalibacterium are included in this phyla. It's colonized a little bit in later infancy, maybe within the first 3 to 6 months. Its proliferation, unfortunately, can be negatively influenced by C-section and early antibiotics, but it's a significant butyrate producer. So is Rosaburia. Lactobacillus, by the way, is in the Firmicutes phyla family. So are staph and strep, which are there normally under normal conditions, but they can overgrow also, especially when there's low stomach acid. So sometimes when we saw the presence of H. pylori, we definitely saw the overgrowth of staph and strep. But the thing is if we don't have that Faecalibacterium and some of the others, the Rosaburia, Akkermansia, which is not part of this phyla, also, they're all butyrate producers. So unfortunately, and next slide, please. There were many of the heroes that had very low or absent butyrate producing Faecalibacterium. So as I said, without the butyrate, we're getting less colonic motility. Had you moved to next slide?
Yes, I can. Yeah, absolutely, Geri. And I also wanted to just do a time check because I know there's so much.
I'm speeding up.
Okay.
I'm speeding up. I know. I think the whole thing we're a little bit behind, and we did leave a nice gracious 40 minutes for questions. But so I think even ideally 30 minutes, or we'll schedule a part 2 for every family. But this is all the critical information. I think so. It's hard to.
I know.
I already abbreviated it.
I know you did. Is this
fine with it all? But anyhow, we got a lot of.
I'll just say that those that are listening, there are going to be questions of, I have no idea what this means. I'd like to just feel free to just exchange this in the chat for all of us, because I'm sure that you're not alone. I certainly have heard this many times and still ask Geri, please explain to me that word or how that actually matters, right? So let's just have a healthy conversation in the chat.
100%. Yeah, that's why I wanted to just try to put as much in the slides, but I have to tell you, I also pulled a lot out, and that's why I called this kind of like first 8 PGI maps round table, because I would think we're all deserving of a part 2. But we are low on our butyrate producing gut bacteria, so which will negatively impact the GI tract's good barrier function, colon motility, and the IgA, which corresponds with that low gut immune response. We wind up with very low IgA. Then that makes the gut more susceptible to inflammation and infections. Okay, so next slide. All right, so as a result of low beneficials in general, we see in many of the heroes what's called an insufficiency dysbiosis, just low beneficials in general. So that is why we want to focus on the dietary recommendations. And again, there can be individual recommendations. You know, should I be meeting with any of you to go over your particular child in greater detail? Because again, your physicians and gastroenterologists, and certainly if you have your own nutritionist who's well versed in this, will be able to guide you. But the GIs and the physicians on the dietary side, remember their role is more on the treatment side, should there have been a parasite of significance or a pathogen of significance that needs to be eliminated. Often those eliminations come with antibiotics, which can cause other downstream repercussions, but the infection does have to be eliminated. So in some instances, so as long as then we put a plan of repair and rebuilding in place, you can pair those approaches for all, you know, all towards good things. So some of these recommendations to build up the good or commensal bacteria. Bina, next slide, please. As you all of these nutrient-dense foods, as represented in the previous slide, also bring with them a lot of micronutrients that help with gut repair, immune system support, neurological support. So I'll speed it up. Next slide, please. Also, they bring with them these prebiotics that help serve to really feed all the good bacteria, which help crowd out the opportunistic or bad bacteria. Next slide. We can also introduce both food-based probiotics or commercially prepared probiotics, of which there are a lot, and we can have a big conversation about that, because some probiotics are a little bit more histamine producing. And if you've got an overgrowth of bacteria that are very histamine producing, and you already have a child who suffers with significant allergies and sensitivities, we may not want to be kicking up their histamine production. So all of this becomes, you know, pretty customized or personalized. As we know, really, nutrition and diet and medicine should be and will be, we hope and pray, in the future. And all of this great research, as ultimately as it will be, as we move from sort of the singular and each individual AP hero that needs attention for gut optimization and health optimization and health span through their lifespan, all of that data that's collected will hopefully go forward and help the collective pediatric, you know, those that are here and those to come. So next page is the opportunistic. So not necessarily the bad per se. They're there. Some are hanging out, but they are definitely opportunists, and they are definitely bad when they are overgrown as opportunists. So because some of them can be associated with great inflammation and with autoimmune disease, as I mentioned earlier, but now you might have a keener sense of how all of that sequela unfolds. So even from anything that reduces stomach acid, and I can appreciate that you might have to put the fire out, but we also then might have to provide some lower down digestive support. Because if your child or you are on an acid inhibitor or blocker or neutralizer for long periods of time, as you suppress that acid, you are impeding the proliferation of the good bacteria and then also allowing the overgrowth of these opportunistic bacteria that then are going to have a negative effect on barrier function and digestion and assimilation and so on. So next slide. So, and when these are very overgrown, we have this inflammatory dysbiosis, right? Again, as I just mentioned. And we definitely had heroes with a lot of overgrowth because, as I also mentioned, we had a lot of heroes that had very low commensal keystone. And we did have some heroes, you know, that presented with candida overgrowth or yeast overgrowth, which can easily happen with antibiotics and also a high carbohydrate or sugar diet, which tends to then feed the candida, which then inhibits the growth of some of the beneficial bacteria and also can exacerbate constipation. Okay, so next slide. Yes, ammonia producing bacteria also can create more disruption. But again, I'm a little getting into the weeds. So moving down, because this is of significance. Yes, there are different types of dysbiosis, and some of the heroes did present with all of it. The insufficiency dysbiosis, not enough good stuff, the inflammatory dysbiosis, overgrowth of too much of the non-good stuff, that led to some of the digestive dysbiosis, the barrier function issues that occurred, and for which, when you see an overgrowth of some of these non-beneficial bacteria, yes, again, they can be represented as SIBO. Some of them are methane producers, some are hydrogen producers. So sure, you might take antibiotics to wipe out all of those opportunistic ones, but unless you really do all the cleanup that we're talking about, make the dietary changes, it will just occur again. So some of the other natural things we can do to help with to inhibit the overgrowth, next slide, please, are some of these different herbs and adaptogens, which have been used for forever, just even curcumin and some of these that are used in spices, and they've been used for health reasons all around the world since the beginning of time. They do have broad spectrum antimicrobial and antifungal effects. Now, it's not about taking all of them. There is some specificity. Again, this is just a broad discussion on this, but just an appreciation of why, especially in warm climates, before there was refrigeration, foods could be heavily spiced, right? Because we even know that certain spices will definitely kill off bacteria. And again, I'll stop there because I'd love to share all the examples with you guys and because it's all great stuff to know. But next slide. So here, which is really the last page of significance, where parasites would show up that should not be there, that will infiltrate a low barrier function, a gut that's very inflamed, and then these intestinal health markers that include ones that look at digestion overall and tolerance and immune response. So let's just actually go down to the next slide, Bina, because we're closing in now. In terms of the parasites, there were heroes where parasitic protozoa were noted. The Dientamoeba fragilis, which is described there, and also the Blastocystis hominis, also described there. And so that's there. We'll just keep moving on, you guys. You know, if it's significant to your heroes test, we could discuss further, but otherwise for general information, it will be on the slides that, yeah, I'm down at the trends observed and nutritional interventions after that on the protozoa, the there were heroes with that parasitic protozoa noted. Perfect. Yep. So if you have questions about that, but otherwise even to read the particulars when you go back through these slides. Okay, next slide, Bina. So the low steatocrit and high elastase under digestive markers is what we want. That would show good digestive function. And most of the heroes did present with this, but there were ranges, right? Many did indicate some low digestive capacity, probably due to some compromised pancreatic output, which could be due to the low stomach acid for some of the reasons that I discussed. Certainly the H. pylori or the proton pump inhibitors, but also stress and poor sleep can compromise digestion and compromise pancreatic output or even stomach acid. So, other markers noted here that I may not have made any mention of, calprotectin or even blood in the stool. Calprotectin is a marker of inflammation in the colon. So those, yeah, you're there. I'm sorry. I didn't know if the slide bounced. So calprotectin as a marker for inflammation in the colon. The secretory IgA, as I already mentioned, it was low in many of our heroes, and it is really that first line of defense to barrier protection against, infiltration into the bloodstream. So many heroes did have very low secretory IgA and also very high zonulin, which is that marker, as I mentioned, in the small intestine. So actually, Bina, if you could move to the next slide with that little more definition of zonulin, it is responsible for what should be those nice tight junctions of the cells of the small intestine that are also responsible for the final stages of digestion. But when we're not getting those nice tight junctions, they're kind of loosey-goosey from all of this barrier infiltration and inflammation, we are going to get that leaky gut, and we're going to have a harder time absorbing our nutrients. And there's, in the next slide, Bina, just a little bit more of a visual in terms of tight junctions. Now, other nutrients affect tight junctions. Vitamin D should always be optimal because it helps maintain those tight junctions. Zinc intake should always be optimal. It helps also with those tight junctions. All right, we're wrapping up. So the additional foods to support the gut microbiome, I'm making mention of the various types of collagen that can come certainly from bone broth, which is a great source of glutamine, but even supplementally, glutamine can be taken, or aloe and other things that can help to provide some support for the rebuilding of that good barrier. Next slide. Again, the lifestyle practices that all go along with it. And the last slide, basically just nice nutrient dense clean foods that kind of lower the inflammatory load that comes with processed foods, and it helps to enhance the fibers and the phenolic compounds and all the good stuff. The last two slides are really just indicators of whether or not there is the presence of antibiotic resistance, and then the other slide being the last slide, to any of within your child's gut, to any of the pathogens that may have been detected. And that can give some insights to your practitioner who may be prescribing something. And then that's it, and that's a wrap. So I'm coming back on. All right. Questions?